Okay, well, it's 10 o'clock and I wanted to go ahead and get started because I want to keep this on time as much as possible. My name is Trent Sutton. I am Justin Priest's faculty mentor, and what I wanted to do is give a brief introduction for Justin for his master's thesis defense seminar. So welcome everybody this morning joining in by Zoom. A couple of quick announcements. If you're on, you may want to turn off your video feed. That can help save a little bit with the bandwidth. We might run into that problem given the number of people that we have joined this morning. And I think Justin muted everybody, so that won't. At the end we'll have a time for question and answer. So we'll unmute everybody at that point, but we'll have you muted for now just to keep down the background noise as Justin is presenting. So just a couple of those quick announcements. Like I mentioned, I wanted to give a quick introduction to Justin. Many of you know Justin well. He grew up here in Alaska, in Hope, Alaska specifically. He spent most of his formative years there and then moved to Palmer when he was in 7th grade, graduated from Palmer High School, then came to the University of Alaska Fairbanks, where he received his Bachelor of Science in Fishery Science from the School of Fisheries and Ocean Sciences and graduated cum laude. While an undergraduate at UAF, he got a number of different experiences along the way. He worked as a gill net deckhand in a commercial fishery. He spent a couple of summers working for Alaska Department of Fish and Game as a fisheries technician in Dillingham and Port Moller, and also in Cordova. After graduating from UAF, he went on to work for LGL, Alaska Research Associates, and he worked on a number of projects throughout the state. And that's where I got to know Justin a little bit. He'd come to campus and recruit students for these projects. And we got to talking a bit about opportunities for graduate school. And I, in 2016, began the project that he's going to talk a bit about, which was the Beaufort Sea Nearshore Long-Term Monitoring Program up in Prudhoe Bay. We took that over. And Justin was a no-brainer to work on that project, given his previous experience with LGL working on that project when LGL was the operator for that. And so over the last couple of years, Justin's been a graduate student working on his master's degree research at UAF on a project he's going to talk to you a little bit about here in a bit. Before he finished up, though, Justin actually now is a fisheries biologist 3 with the Alaska Department of Fisheries, Alaska Department of Fish and Game, Comfish Division, working down in Sitka. So he's done some pretty amazing work on his project, and he'll tell you a bit more about that. But before we get to that, I wanted to, we all know those sort of basic things about Justin, but there's some things we also don't know much about Justin that through conversations I've had with him and my exhaustive research into his background, I've been able to unearth many of the things I'm going to reveal today. Most people don't know. As a matter of fact, I think I might be the only person that knows some of these things. So I like to kind of call this Justin Priest, the untold story. What most folks don't know is Justin's aspirations really initially weren't in fisheries biology at all. He actually wanted to be a reality TV phenom of sorts. He wanted to gain fame and fortune in reality television. And so his first actual attempts in this regard were he wanted to be on a reality TV show, and his actually dream show he wanted to be on was American Chopper. And many of you know a bit about this show. It's and Justin tried a number of times to become one of the individuals on the show. And it wasn't so much that his personality didn't fit very well. It was more that he couldn't quite get the hairstyle, the facial hair down quite right. He tried. I mean, you see that. He's definitely trying. But it just wasn't quite what the show was looking for. So his attempts to get cast on the show as one of the individual participants didn't quite work out so well. His next attempt was to create his own reality TV show. And talking to some of his friends, they decided to model one after a show that many of us are familiar with. That's Alaskan Bush People. If you're from Alaska, we unfortunately know about this show. Unfortunately, the take that he had on this with his friends, called Alaskan Caribou People, didn't quite have the same cachet as Alaskan Bush People. It didn't quite take as much take off like the other show did. So they didn't quite get where they wanted to go with that one. So put that on hold. But really, I think Justin's claim to fame and really kind of was sort of a man before his time was some of his interactions earlier in life. And where I say he kind of led to something that's become quite big today is, I think we're all familiar with the show Tiger King. Unfortunately, I'm familiar with the show Tiger King, which has become this sort of hit reality TV docu-series involving Joe Exotic and his trials and tribulations as a zookeeper in Oklahoma with tigers and interactions with tigers and other big cats. Justin has no interaction with tigers whatsoever, or any big cats for that matter. But where he kind of comes in this is some of his earlier interactions. And again, maybe this is a function of bad timing or poor marketing, but his attempts that kind of laid the groundwork for Tiger King just never really took. And that was an episode of a show he tried to put together called Marsupial King. And it just didn't kind of work out the way that Justin wanted there in his interactions with kangaroos. But nevertheless, again, it may have laid the groundwork for the Tiger King that we know so much about today. But what really came out of all these different attempts to become a reality TV star, Justin realized that wasn't his forte. And so through these attempts, he kind of learned kind of what he wanted to do in life. And that kind of led to the project he's working on today, and where sort of his strengths are, which includes sitting in front of a computer and playing with data. So with that, I turn the floor over to Justin, and I'll stop sharing. So Justin, there you go.
All right. Wow. Well, thank you for that, Trent. I see you dredged the best and worst of my Facebook for all that. That was wonderful. So anyways, well, thanks everybody for coming today. Today, I'll be talking about some of the research I've been doing over the last too many years for my master's, looking at the effects of environmental variables on nearshore Arctic fishes. Give me 1 second. So first, I'd like to acknowledge that this is a collaborative effort with substantial help from my committee members, Trent Sutton, Franz Muter, and Scott Rayborn. Hillcore Alaska provided the funding for the study, as well as supporting my graduate research through the UA Foundation. Before we begin, I'll mention a little of the motivations behind my project. As most of us have heard, there are substantial changes occurring in the Arctic. Sea ice has been rapidly declining over the last 4 decades with unknown environmental impacts. In addition, there have been substantial increases in average water temperature. This plot on the right is from our project. So these environmental changes led me to ask what sorts of impacts there would be on the local fish community. Often one of the first things I'm asked is why there seems to be more warming occurring in the Arctic than in lower latitudes. To demonstrate this, I've made this little cartoon showing the phenomenon called Arctic amplification. In the first panel, there's a lot of sea ice covering the ocean, and sunlight for the most part is reflected off the surface. In the second panel, some heat has caused cracks in the ice. Now more sunlight can enter the water column. In the last panel, we see that less sea ice has now caused even more heat to enter the water column, leading to even less ice. Thus, we see that more heat there is in the Arctic, the faster that melting occurs, which leads to even more heat. A second phenomenon occurring in the Arctic regards the amount of sunlight entering the water column. It was theorized that a reduction in sea ice coverage would mean more light in the ocean. In the first panel, we see that sea ice is bound hard to shore and covers the surface. The reduced sea ice in the second panel has let in more sunlight in the water column, leading to an increase in primary and secondary production. However, recent research has contradicted this and refined our understanding of the Arctic. Instead, what we see is that a reduction in the shore-bound sea ice has increased the amount of wave activity, causing an increase in erosion. This erosion causes suspended sediment in the water column, blocking light, reducing production. Erosion is happening in the Arctic at almost unbelievable rates. In most locations, there are 1 to 3 meters lost per year, but up to 25 meters per year in some locations. While the study that I'm going to present on today did not address turbidity, as with any data set, it's worth keeping in mind that there are longer-term trends not captured in the analysis. So when we look at the changes over the years, it represents a whole suite of changes that are occurring. This study takes place along the Arctic Ocean in the semi-estuarine environment of Prudhoe Bay, Alaska, off the Beaufort Sea, denoted here by a star. Right near Prudhoe Bay, the Sagar-Venurktok River enters the Arctic Ocean. Other major rivers in the region include the Colville River, 80 kilometers to the west, and the Mackenzie River, 500 kilometers to the east. This entire region of Alaska is commonly known as the North Slope. If you have heard of Prudhoe Bay, it's because it's a major hub for oil and natural gas extraction, shown here. There are substantial industrial buildups in the areas right around the Arctic Ocean. These industrial activities occur just feet away from pristine Arctic habitat. The infrastructure for the oil production also make this one of the most accessible locations for studying Arctic waters. Prudhoe Bay isn't only unique because of all the industry. The nearshore region is extremely shallow, with the 2-meter contour hundreds of meters or sometimes even several kilometers from shore. These shallow waters also have minimal tides. In place of tides, water height is driven by wind direction. The wind can change the water height by up to 2 meters, or approximately 1 kyle, as shown here in the top left. Nearby barrier islands and the mouth of the Saguenay River create semi-estuarine conditions, ranging from fully marine to completely freshwater. Finally, the other extremely unique feature is the presence of sea ice from early October until June, though these dates have been in flux lately. Again, here's Prudhoe Bay in context, represented by this star. Sampling took place at 4 sampling stations near Prudhoe Bay. From east to west, these stations are named Site 230, 214, 218, and 220. The colors on this map will represent each of these stations throughout the rest of this presentation. You can notice immediately that there are 2 stations present in the east near the mouth of the delta, represented by pink and green dots, and 2 stations in the west, farther from the river mouth, that are blue and orange. So the uniqueness of the Arctic and the changes that we've seen led me to ask my overarching research question. Given these changes in the environment, are there also changes in the fish community or in individual fishes? To answer this question, I split the problem into 2 chapters. Chapter 1 focused on how the environment affected the overall fish community, so looking at changes in all of the species. In contrast to the overall community approach of Chapter 1, Chapter 2 took a more species-specific approach. This chapter focused on just the effects of water temperature and salinity on the juveniles of 2 whitefish species. By the end of this seminar, there's a few things that I'd like you to hopefully take away. First, in Chapter 1, we'll find that Arctic fish community is undergoing changes, such as an increase in species richness, which possibly benefit generalist species. Secondly, these shifts in the fish community are tied to changes in the environment, especially those driven by climate change. Moving on to Chapter 2, we will learn that the growth of juvenile whitefish is increasing with warming conditions. We'll also learn that changes in the environment significantly affect the abundance of juvenile whitefish. And lastly, a little side note. Throughout this talk, I'll be using the term CPUE. CPUE is simply catch per unit effort, or the amount of fish caught accounting for the amount of effort that we put forth. Catches in the CPUE will be assumed to be proportional to the relative abundance of fish. So now that we've seen that this is a very unique environment, why should we even care about Arctic fish? Well, first, as the Arctic warms, there have been documented shifts in northward moving distributions of fishes in the northern Bering Sea and southern Chukchi Sea. So one research interest of mine is whether we could detect an influx of Pacific fishes coming into the Arctic that could displace or affect the abundance of Arctic fishes. Secondly, many species of Arctic fish are very important sources of subsistence foods for local villages. Many of these people are the local Inupiaq who depend on food for livelihood and culture. Lastly, fish are very important as a keystone species for higher trophic species such as beluga whales or mirrors. These species then in turn support much of the entire Arctic ecosystem. To study how nearshore fish communities are changing in the Arctic, we would need a long-term data set. This is because the Arctic is naturally extremely variable. Luckily, we have a long-term monitoring project where sampling has a complete data set since 2001. While sampling has occurred at these locations since 1981, these data were not digitized and so were not able to be analyzed. All analysis was performed on the data set from 2001 through 2018. Sampling occurred at 4 standardized spike net stations with a double-ended net set 60 meters from shore, as shown here. This was to capture fish moving in either direction. Sampling was conducted daily at each location from approximately July 1st through September 1st, contingent on safe access of the site and sea ice conditions. During all sample events, we collected data from fish by looking at total counts for all species captured. For a selected group of species, we also took length measurements. Environmental data were also recorded during every sample event. We used a YSI to measure the water temperature and salinity. Here's a diagram summarizing some of my overall workflow. The 18 years of data from the project were all in different files, which needed to be cleaned up and standardized into a single file. These data were then imported into R, where I used a variety of packages to clean up the data. Analysis was done using several packages, including MGCV and Vegan. Visualization was all performed using ggplot2. And finally, these data were pushed to GitHub on project-specific repositories shown here along the bottom. As a quick summary of the overall project data, I made this infographic. Data analyzed were from 2001 through 2018, during which protocols remained substantially unchanged. Sampling occurred during all 18 years from approximately July 1st through September 1st. This gave us a total of 995 days to analyze. We captured 33 different species, which spanned a range of diadromous, marine, and freshwater fishes. In total, there were over 425,000 lengths which were measured during this time frame. The data were analyzed against up to 4 environmental variables, which were water temperature, salinity, Sagavanirktok River discharge, and wind. The total number of fish captured during the 18 years of the project were approximately 1.8 million fish total. All right, so that brings us to chapter 1. This chapter looked at how the fish community has changed over time in response to environmental variables. This chapter's research was divided into 3 objectives. The first objective that I wanted to look into was the effects of environmental variables driving trends in species composition. Secondly, if there were trends, I quantified how species composition, abundance, and variability changed over time. And last, I investigated the temporal patterns in both species richness and the presence of rare species. First, all analysis was done on a biweekly scale. The bi-weeks were split according to the timeline by dividing the season in fourths, approximately 2 weeks long, which I'll refer to as bi-weekly periods. To prevent rare species from overwhelming the analyses, we excluded these species. So we only analyzed species that we caught more than 100 fish of in all samples and years combined. This left us with 17 species to analyze. All of the CPUE and environmental variables were transformed and standardized so that everything was on a comparable scale. Finally, for the multivariate analyses, we used a Bray-Curtis dissimilarity of the CPUE. This is just a way to measure how similar or different each sample point is from another in higher dimensional space. As mentioned before, we had 4 environmental variables that we assessed. Water temperature and salinity were both measured during the course of this project during every sample event. The east-west wind vector was collected by the National Weather Service, and the Sagavanirktok River discharge data was from USGS. These environmental variables were assessed in a multivariate framework. Before we analyze the fish community, I'll show you results from the environmental variables. The plot on the left shows salinity over time, with each station its own color. We see that there are clear differences by station, but no distinct overall trend. For temperature on the right, there's clearly a difference by station as well, but also a warming trend. Testing for significance, we see that salinity had no change over time, but temperature significantly increased by approximately 1.4 degrees Celsius over the study period. For Saginaw-Vanderktok River discharge, we see that there's also a significant increase in discharge over time. For the wind vector, we see that there has been a significant increase trending towards stronger easterly winds over time. The species that were captured were reflective of the estuarine environment, with several families or subfamilies being commonly present. In most years, whitefish, as shown here in blue, were the most common group caught, ranging from about 5 to 75% of the catch. In some years, there were also occasionally massive catches of cods, predominantly driven by Arctic cod. Because I'll be talking a lot about species composition, I wanted to define it first. Species composition, or sometimes I'll use the term assemblage structure, is simply the proportion and abundance of species. Species composition can change in several ways. In this first comparison, we see that time 1 is different from time 2 because the abundance has doubled. For the bottom groups of fish, we can see that the proportions have now changed. For our analyses, we want to determine changes in either the overall proportions between species or in the overall abundance for all species at once. But how can we test the trends in species composition when it's made up of dozens of species and detects changes in either proportion or abundance? To look at all these variables at once, we'll need a multivariate approach. This multivariate approach is the basis of my first objective. I looked at what the effects of the environmental variables were on the species composition. To do this, I used a non-metric multidimensional scaling, or NMDS, as well as a permutational analysis of variance, or PERMANOVA. Now, a word of warning for the multivariate analyses of objective 1. Because we'll be assessing everything in multivariate space, we won't be able to precisely quantify magnitudes of how environmental variables affect the species composition. First, I looked at the total species composition and did this using a non-metric multidimensional scaling plot. The NMDS plot was best fit with 3 axes, but for the sake of time, I'll only display 2 of them here. If you're not used to NMDS ordination plots, they can be a little confusing at first. Every sample is its own separate point, so all the proportions and abundances for all the catches of the species are combined into a single point. What's important here is not the precise values, but rather how close together or clustered different points are. For the NMDS ordination, each sampling location is a different color in this plot. You can see that the blue and orange colors are found together in the top part of the plot, and the pink and green are in the bottom part. Putting the 95% confidence interval over the plot, we see that indeed the centroids of the stations are distinct. So there's significant catch differences between some stations. You might be wondering if there's a reason for this order. Going back to the map, we see that the pink and green stations are in the east near the river mouth, while the blue and orange are in the west. Keeping in mind their locations, it's clear now why each of the colors would be similar. Going back to the NMDS plot, we can see that the western sites and eastern sites both ordinate out together. Okay, so catches are different between sampling locations. What about how the environmental variables affect the species composition? To look at this, I quantified the relationship between these environmental vectors and the NMDS data points shown here. You can see that the salinity vector is longest and it has the highest impact. It also goes perpendicular to the blue and orange dots, indicating that these stations are strongly correlated with salinity. Biweekly period was also important, but did not appear to be associated with stations. The environmental variables of salinity, biweekly period, temperature, and year were all highly significant. Discharge and wind did not significantly affect the species composition. Next, we overlaid the top species associations over the plot. We see that certain species are associated with different stations. For example, capelin up here with the blue and orange dots is highly positively associated with salinity. While in contrast, we see that round whitefish and Arctic grayling are negatively associated with salinity and found more at the eastern stations. Next, I looked at the effect of environmental variables by performing a permutational analysis of variance, or PERMANOVA. The top PERMANOVA model had a significant associations between the species composition and all 5 variables. Just including salinity and biweekly period accounted for over 1/3 of the explained variation in catches. The top model explained 47.5% of the sample variation. Thus, there's a high level of variability in catches that's unexplained by local catch conditions. After this, I looked at which species were driving a lot of the changes in the catches. I did this by performing 2 separate analyses. First, given that most of the differences are between the east and the west sampling stations, I investigated which species were driving these differences by running a similarity percentile, or SIMPER analysis. The SIMPER analysis showed that the top 3 species accounting for the differences were round whitefish, 3-spined stickleback, and Pacific herring. Several of the other species rounding out the top influencers were diadromous and also generalists, but aren't shown for space. Separately, I ran Pearson correlations between all species and the 3 NMDS axes to identify the dominant species affecting species composition. 3-spined stickleback was the species most associated with axis 1, while round whitefish was the species most associated with axis 2. Note the general agreement between these 2 different analyses. We see that both approaches agree on the top species driving changes. The takeaway here is that the species that are mostly driving the changes are diadromous and/or generalists. For example, round whitefish and 3-spined stickleback are both tolerant of a wide range of environmental conditions in the estuarine environment and able to move back and forth between marine and freshwater conditions. For the second objective, I wanted to quantify the seasonal and interannual trends in 3 things. Again, looking at species composition, but also looking individually at abundance and variability. The analyses here still only looked at the common species and excluded the rare species. Examining whether there were any trends in the NMDS axes, we fit GAM models to the data points from all 3 NMDS axes. As a reminder, MDS is just the axis points from the NMDS chart shown earlier. We find that NMDS axis 1 has a significant trend over time. Similarly, MDS2 also displays a significant change over time. MDS3 did not display a significant trend and is not shown. The trends in NMDS mean that the species composition has significantly been changing over time. Again, since we're looking at the species composition from a multivariate composite, it doesn't necessarily matter whether these are increasing or decreasing. What does matter is that we can see that there are significant changes in the axes over time. As I mentioned, our results so far have been multivariate, which showed that there was change, but wasn't able to quantify changes. Since I found significant trends in the multivariate species composition over time, and as a function of environmental variables, I assessed the change for each species in a univariate linear model. For this model, I use CPUE as the response, predicted as functions of year, station, bi-week period, water temperature, and salinity. The species shown here are the 5 species that significantly changed in abundance and were associated with water temperature or salinity. For example, both broad whitefish and saffron cod significantly increased and are positively associated with temperature. Conversely, humpback whitefish, four-horned sculpin, least cisco decreased. While salinity did not significantly change over our study period, it is strongly tied to abundance of several of our species. One thing that pops out is that the 2 species that are increasing both seem to be generalist species. Both species are tolerant of a range of acceptable temperatures and salinities, as well as being able to cope with increasing temperatures. Finally, I tested whether a variability of abundance has increased over time for each species. The rationale for this was that stressed ecosystems often have quite variable abundance. This can be a harbinger of a regime shift. However, in this study, there was little evidence of an increase in variability of abundance. We did see that there was an increase in variability for Dolly Varden catches and a decrease in saffron cod catch variability. I did not perform a Bonferroni correction. The final objective for this chapter was to quantify some of the temporal patterns in species richness, as well as rare species presence. The analyses for this objective did not exclude the rare species. The first analysis for this that I did was to see how species richness changed on an intra-annual as well as interannual basis. The modeled species richness was best assessed using a generalized additive model. I found significant increases over the season during each biweekly period. So species richness is typically highest later in the season. The interannual species richness also increased, yet in a linear manner. We find a significant increase in species richness over time at the rate of approximately 1 species per decade. This surprising finding is not necessarily the result of any new species to the study area, but rather an increase in the species that have already been caught in the Arctic. Intraannual species richness was highly correlated to salinity, as shown in this animated GIF. The interannual week is along the x-axis for both plots. For the plot on the left, species richness is along the y-axis, while for the right, salinity is. Each frame of the animation is a different year. You can see that the curves on the left and right are more similar than different within years. When salinity increases, so does species richness, and they often even arc in the same way. In certain years, the curves line up to almost match each other. This shows there's a high correlation between salinity and the number of species caught. So because we threw out so many species in all the modeling before this, we needed to look at whether there were any trends in the uncommon species as well. To do this, I performed a binomial presence-absence model for each of the rare species. 3 rare species showed significant changes over time. Bering cisco has significantly decreased in probability of occurrence, while burbot and slimy sculpin have both increased. To summarize what we've seen for chapter 1, we've seen that the overall community shows evidence of some shifts, with significant differences among stations and across time. There's also been an increase in the number of species annually caught, with salinity and to a lesser extent temperature proving influential to species composition. Finally, we see some evidence that favors generalist species. All right, I've been talking for a while, so I need to take a breath and get some water. Otherwise, I feel like I'll end up like the guy on the right. All right, now that we've seen that there are some changes in the overall fish community, especially in relation to water temperature and salinity, let's examine the effects for 2 important species. That's the focus of my second chapter. The research for this chapter looked specifically just at 2 species of whitefish, Arctic cisco and broad whitefish. Whitefish are related to salmon, and similar to salmon, they're born in freshwater and spend time outside their natal rivers. Juveniles of both whitefishes rear in nearshore waters of the Arctic. Notable differences between these 2 species are that Arctic cisco are more pelagic feeders, while broad whitefish are demersal. Juvenile Arctic cisco also seem to range farther from shore, while broad whitefish seem to be a little more homebodies. Lastly, note the different colored circles around both of these fish. For the rest of this chapter, I'll display Arctic cisco in green and broad whitefish in orange. So why should we even care about either of these species? As mentioned at the beginning of this presentation, both species are important subsistence resources for the people that inhabit the North Slope. Much of the harvest also comes during late winter with under ice set nets. This provides food during a time of year when food is hard to come by. This has meant that these species are celebrated in Inupiat culture, even being a key ingredient in aguirre, an Inupiat delicacy. Because of the value of these species to local people, both species have been the focus of studies in Prudhoe Bay, including the historical data from this project. A little bit of background on how water temperature and salinity should be expected to affect growth. For most species, we would expect an approximately normal distribution of their response, shown here. As the environmental gradient increases, there's an optimal niche for each of the species. If it's too hot or too cold, we'd expect the response to reflect that. I call this region the Goldilocks zone, as it's just right for the fish. For both of these study species, It's unclear where precisely we are on this curve. Also note that if we're on one side or another of the curve, we'd expect the response to appear to be approximately linear. So why would relative abundance or CPUE be affected? Well, we would theorize that mobile fish, such as these species, would move where the conditions are best for them. Some more background on these species is their distribution. For broad whitefish, we see that they are broadly distributed across the Arctic, if you'll forgive the pun. Many of the Arctic coastal rivers support populations of broad whitefish. In contrast to broad whitefish, Arctic cisco in Canada and Alaska are all immigrants from Canada. There are no known spawning populations in Alaska. Instead, all of the fish in Prudhoe Bay travel more than 500 kilometers, pushed along by wind-driven currents. After rearing for several years in Alaska, once Arctic cisco reach maturation, they return to the Mackenzie River. That brings us to my research objectives for Chapter 2. We had two objectives which were related. We wanted to see how water temperature and salinity would affect biweekly growth rate and also relative abundance. This was done for two age classes of juveniles, age 0 and age 1, and also for both Arctic cisco and broad whitefish. As a review of the annual trends discussed in Chapter 1, we see that water temperature significantly warmed by about 1.4 degrees Celsius. There was no significant annual trend in salinity. On an individual season, however, water temperature cools and then also becomes saline. These trends are potentially caused by seasonality in offshore marine upwelling. Because of these trends, we'll need to account for biweekly period during any modeling so that we can control for these effects. Because this project had so many length samples, but no otoliths from every year, we had to use the length data to determine likely ages of the fish. To do that, we needed to determine which lengths belong to which age. The cutoffs between each age varied over the season and also substantially between years. In a given year and species, we expected that the lengths for each day would have several normal distributions, one for each age. Over the course of the season, these distributions would increase linearly. These distributions and growth of the season were evaluated using a mixture model for each species and year using the formula shown below. Let's look at how the mixture model would work using broad whitefish lengths from 2017. In this chart, we see day of year across the x-axis and lengths across the y-axis. We can clearly see there are several clouds of ages of fish that separate out. The age 0 fish are the smallest group, with clear breaks for ages 1 and 2 as well. Applying the mixture model, we find the length breakpoints between where a fish is more likely to be one age than another on a given date. And voila, we can now apply this model to all of our lengths. It should be noted that these age delineations agree with otolith ages currently being conducted by other members of my lab. Now that we've assigned out ages based on fish length, day of year, and species, we can investigate some basic trends. We see that in this chart of abundance over time by species and age class, that the abundance is quite variable. Many times the annual abundance is quite close to 0, though we see large spikes in other years. Looking at how mean length has changed since 2001, we see highly significant increases in mean length over time for all 4 groups. This increase in mean lengths lines up with previous research in the region. Also note that Arctic cisco mean lengths, shown in green, were consistently higher than broad whitefish. Once we have the likely ages for each length, we can determine the growth over a biweekly scale. Growth was determined using a linear mixed effects model, which allowed for unique slopes and intercepts for each species, age class, year, and bi-week. Growth is simply the slopes for each of these biweekly periods. We'll use growth as the response variable for further modeling shown in the formula here. Next, we'll be able to investigate why there are differences between the biweekly growth rates. For example, why is one certain bye week have a higher growth rate than another? So now that we have growth measured for each species, age, year, and bye week, we can model what factors affect growth. This was modeled using a generalized additive model to account for nonlinear curvature, and top models were selected using AIC. First, we'll look at the results for water temperature. All 4 groups showed a positive linear trend for the effect of water temperature on growth rate. We see that comparing between species for age 0 fish, that the effect of temperature on growth is 3 times greater for Arctic cisco than broad whitefish. Comparing between age 1 Arctic cisco and age 1 broad whitefish, we see that the slopes are essentially the same, meaning that water temperature had a similar effect between species. Now for the same modeling, but looking at the results for salinity. Model selection revealed that only age 0 Arctic cisco were affected by salinity. In this model, we see that there is a negative linear relationship between salinity and growth. This negative relationship agrees with previous mortality research by DeMarch that showed a similar general trend. Moving on to objective 2, I modeled CPUE to look at how water temperature and salinity affected the abundance of juvenile whitefishes. If you recall back to the abundance charts, there were frequently 0 catches in many of the bye weeks. This meant that we would need to use a negative binomial distribution to accurately model it. We also needed to account for some of the temporal trends. To account for the differences among stations, we'd use a mixed effects model with station as a random effect. So putting all this together, I used a negative binomial generalized additive mixed effects model. To determine top models for this, I again used AIC for model selection. The results from this model fit the data well. Here's the predicted catch for each station and during each bye week. Each frame of this is one of the 4 different bye weeks, as shown in the bottom right. We see that the catches of age 1 Arctic cisco in dark green and age 1 broad whitefish in dark orange are fairly consistent between stations and bye weeks. There's a large catch of age 0 fish in lighter colors in the last bye week, especially of age 0 broad whitefish, shown in light orange at these delta stations. Looking at the results from the top models, we see that AIC model selection showed temperature to only have an effect on CPUE for age 1 fish of both species. For Arctic cisco on the left, we see that there is a U-shaped curve in predicted abundance in response to temperature. For broad whitefish, we see a linear increase in how water temperature affects abundance. Last, we look at how salinity affected the CPUE of the juvenile whitefish. The top models from AIC selection showed that salinity only affects age 0 and age 1 broad whitefish. For age 0 broad whitefish, we see a nonlinear curve with CPUE maximized around 10 parts per thousand salinity. For age 1s, we see a negative linear relationship between CPUE and salinity. This study found that increases in water temperature and fluctuations in salinity led to changes in growth and CPUE for both age classes of juvenile whitefishes. Taking together the results from modeling growth and CPUE, it appears that the ongoing trend of warming nearshore waters has enhanced rearing conditions for juvenile whitefishes. For other whitefishes for which this research has been done, it appears that maximum growth occurs as high as 18 degrees Celsius, far warmer than current Arctic conditions. So, for example, we don't quite know where we are on this curve. Also, just because their growth might increase, it isn't clear how their prey would also respond. Perhaps some of the ice-associated prey are negatively affected at warmer temperatures. Lastly, in a changing environment, not everyone can be a winner, and we might expect that some species might have optimal conditions at the expense of other species. It's not clear how increased growth and changing abundance could affect other species. Now that we've seen the results from this research, I'd like to put these findings together and provide some context. So just to review the overall findings from Chapter 1, we found several very important results. First, we saw that there were significant changes in the environment occurring, most notably an increase in water temperature. Second, I determined that there were shifts in the overall species assemblage structure, though because these were multivariate analyses, we could not determine the precise amount. Many of these changes in fish abundance and proportions were associated with the previously mentioned changes in the environment. I also found that there was a significant increase in the number of species in nearshore waters over time. This was increasing at approximately 1 additional species per decade. Lastly, I determined that much of the changes that were observed seem to indicate trends towards species that are generalist species. These generalist species are able to exist at a wide range of conditions and tolerate the existing changes that are occurring. For chapter 2, we looked at growth in CPUE for age 0 and age 1 Arctic cisco and broad whitefish, summarized here. I found that water temperature positively affected the growth of all species and ages. For salinity, my results showed that salinity only affects the growth of age 0 Arctic cisco. For the models regarding relative abundance, water temperature affects CPUE of age 1 Arctic cisco and age 1 broad whitefish. I also found that salinity affects the CPUE of age 0 and age 1 broad whitefish. To put these two findings of chapters together, first, we've seen today that changes in environmental conditions, especially climate change warming, are driving shifts in the Arctic. We also documented shifts in the Arctic fish community, for example, increasing species richness, possibly benefiting generalist species. Looking at individual species, we found that growth in juvenile whitefish is increasing with warming waters, and that juvenile whitefish relative abundance is affected by changes in environmental conditions like water temperature and salinity. These findings show that fish in the Arctic are changing on both the community and individual species scales. What we don't quite know, even with all this research, is how exactly to place all this in the proper context. We're left asking, what does this all mean? Looking forward to future research, my first and probably most obvious point is to continue the existing monitoring program. When projects are started, you often don't know where it will lead, but the value comes through keeping the time series going. Other studies could involve other species in the nearshore. Fish are just one link in the overall Arctic ecosystem. What happens to the trophic levels above and below them is extremely important in determining what their future will look like. Also, as mentioned earlier, we don't know that everything in the ecological web will move in tandem. We don't know how fish prey and predators will respond to different conditions in the Arctic. Long-term nearshore fish diet studies would also be extremely valuable. This situation is currently under study. Unfortunately, I was not able to assess either sea ice extent or turbidity due to a lack of data. Determining nearshore sea ice presence and extent requires processing high-resolution imagery that is currently being analyzed by Dr. Andy Mahoney at UAF, so this might be an option in the future. Similarly, this study did not quantify the effects of turbidity due to not having collected these data. The changes that we've seen that are occurring will likely continue into the near future. My research has shown that there are definitely changes occurring, but there isn't yet a regime shift. Given a future that looks warmer and rainier, we just don't know how the fish community and individual species will respond to the predictions of a warmer, less saline nearshore. We're also pretty sure that the Arctic will continue to warm, and we've shown that growth will likely be high for the species we've looked at. What we don't know is whether this growth is a good thing. Often growth is beneficial to fish survival, but sometimes the opposite is true. If the growth does not occur in line with how the species expects conditions to be, that can actually have a very detrimental effect. One common theme running through this study is that generalist species seem to be positioned to do better in a changing environment. Like the 1960s horror film poster to the right, this might seem to be a little scary. However, like the campy films, it might end up being a situation that isn't necessarily to be feared. I'd also just like to highlight that we don't quite know what the future will bring. which is to say, we don't know how specialist species will fare against generalist species. As the Arctic becomes more like lower boreal latitudes, we would expect that we'll see new species in the future. We didn't necessarily see any new species that were new to the Arctic in this study, though we did catch a sockeye in 2017, a first for the project. One fish doesn't a trend make, but what happens if we start to see fish from lower latitudes of the Pacific or Chukchi show up? The species that show up would, by definition, likely be well suited for future conditions. The only option for existing nearshore species is to adapt or to die out. And with that, I'd like to acknowledge folks. The only reason I've been able to accomplish anything has been because of the amazing support from an amazing number of people. Whether it was colleagues and advisors that I turned to for help when I was really, really, really stuck and felt like this, by either just being a sounding board or providing me with actual code to run, it was much appreciated. Or whether it was my family and friends for those moments when I needed to vent in order to stay sane. Thank you. So bear with me in taking a moment to try to thank some specific people. So many people have helped me with this project over the years. Philcor Alaska and Beth Sharp provided the funding and overall project management for this research. Christina and Gabby helped me with all of the admin things. The great staff and faculty of UAF Sitka helped make me feel at home while away from a UAF campus. AFS Alaska awarded me funding to attend conferences to present, discuss, and hone my research. In addition to the many people that have helped with the project, so many more people have helped guide my path as a biologist, both before and during graduate school. I wish I had time to thank each one of these people individually and say how awesome they are. So to each of you, thank you. I heard this quote when I first started grad school. There's two things you never want to do: start a long-term monitoring project and end a long-term monitoring project. With that said, I'll need to thank the many people that have been involved with this research. With this long-term study, there are so many people that have taken turns carrying the torch. I put this many names up there just to show how dependent we are on the dozens and dozens of people that work before us. Along the bottom is a reverse chronology of technicians who have worked the project since 2001. You might notice a familiar name there in the middle. I never thought working this project in 2008 as a young technician that it would come to be so important to me later. It's humbling to be a part of this legacy. Many sincere thanks to everyone who helped push this project forward over the years. I really have to thank my mom, dad, and brothers. It's cliche to say, but they're honestly the reason I'm here today. Each step of the way, they've supported me to become a better scientist in person. Whether that was driving several hours to watch me give my first talk at a conference, taking me fishing, or just listening when I needed it. I've been so focused on my research, I didn't call often enough or spend as much time together as we both wished. Thank you. Thanks also to every single one of my friends over the years for the support, laughter, and the shoulder to lean on when I needed it. Special thanks to my lab mate and field partner, Duncan Green. His approach to demonstrating how to stay safe in the Arctic Ocean, his special Arctic field fashion while sampling, and all around great humor was much appreciated and resulted in many improvements to my research. Also thanks to Kyle Gatt. Whether it's getting skunked at ping pong, staying all smiles during the daily pumping up of our boat, affectionately known as Floppy, or showing the proper technique of how to present your poster at the National AFS Conference, Kyle's attitude was key to me making it through some tough field work. Together, I owe both of these men a debt of gratitude for all their help over the years, even if Duncan can't seem to keep his clothes on, and Kyle seems to wear the same thing in all my pictures. My partner, Rhea Ershman, where to begin? To my patient reviewer, editor, code fixer, idea bouncer, talk me at the ledger, and every major and minor role in between, I'm really, really thankful. Thank you for laughing at my dumb jokes when I was at my best and putting up with my crap when I wasn't. Finally, I'm extremely appreciative for the support and guidance from my graduate committee. Much feedback, manuscript review, and improvements to statistical approaches were thoughtfully provided by both Dr. Franz Buter and Dr. Scott Rabel. I can't tell you how impressed I was to get high quality advice with such quick turnarounds. Many sincere thanks to both of you. My final thank you is for my graduate advisor, Dr. Trent Sutton, who was consistently understanding while providing mentorship, even if he made his own students take him grouse hunting. Sometimes I accidentally took advantage of this, but Trent gave me enough rope to learn so much over the years. He challenged me to improve myself and my research, taught me how to critically evaluate my ideas, and guided me when I needed it most. He's also one of the funniest people I've known, and I'm very happy my path to grad school led me to his lab. I swear I'm almost done. Lastly, thanks to everyone for listening today and joining. I just have one last quote that really resonated with me, so I hope you'll appreciate it as well. It is a country of great variety, enhanced by dramatic seasonal opposites, of light and darkness, warmth and cold, frantic growth, and ice-sealed dormancy. I don't know that I've ever been anywhere with a stronger sense of place than the Arctic. It's a land of many possibilities and extremes, forever changing, but hurtling towards a future out of its control. Thank you to everyone for listening today. It's your interest in the Arctic that can help save it. So with that, I'm happy to take any questions. Hold on.
Okay, we have some time for questions for Justin. So how do you want to handle this, Justin? Just people.
Yeah, if you have a question, just go ahead and jump in. We've let everyone be able to unmute themselves. Or you can write in the comments and we'll call you out by name. Oh, Rich says you're still muted. How do we unmute?
I can hear you fine.
I think you should be able to unmute yourselves. If you have a question, I can manually unmute you. Oh, Christina has a question. I'll try to find Christina.
Or you could also, if you put questions in the chat box and we can read them off that way.
Yeah. All right. Christina, I can unmute you if you want me to unmute you right now. So just be careful. I'm going to unmute you, Christina. There you go.
Hello.
Yeah.
Hi. Way to go,
Hey.
Justin. That was great. It was so good to see the results of all the work you've been doing over the years.
Thanks. Yeah.
I had a question. When you're talking about how the generalists are like winning, basically the rise of the generalists. Does that also refer to species that are more generalist feeders, like generalists in their sort of trophic niche, or is it more in their environmental niche?
You know, that's a really great question. I didn't actually ascertain the difference between those. So for me, what I was looking at was mostly the difference between whether their response to environmental conditions. So it was less to do about trophic niches and more about, I guess, environmental, how well they can handle their environment.
Sure.
So, but that's definitely something I would like to look at more. I did look at whether there was differences in functional diversity. So that differences with species that overlap in their trophic niches.
Right.
But I didn't quite see too much in differences there.
Okay, cool. Thank you.
Yeah, thanks. Let's see, Molly Payne, I see has a question. I'm gonna unmute you here, Molly. Or actually, I'm gonna click unmute all. I hope that doesn't mean everybody's clicked. Okay. All right.
Okay.
There we go. All right, now everyone should be able to unmute themselves, I believe. I don't know.
Can you hear me?
Yeah, yeah.
Hi, Justin. It's my name's Molly. I've never met you, but I learned a lot about you in this talk, which was cool. That was a really great talk. My question was, so you're saying that generalists are increasing and are going to continue that trend, and you're also seeing increased species richness overall. I'm wondering, like, really long term, if you think there's actually going to be a trend of maybe like generalists increasing, but then all other species decreasing. So maybe decreased species richness overall if generalists are going to fare better in a drastically changing environment.
Sure. Yeah, that's a fantastic question. There's kind of like a twofold push and pull there with that. I don't know. I actually don't know the answer to that question. So one thing that I've talked about, and this is definitely a hat tip to Duncan Green here. While we were working, we talked a lot about how if the species keep increasing their northward distributions, then you might see there's a turning point, right? So if you think of Alaska in the north as shaped like a square, once they keep going north, then all of a sudden, once they reach Barrow, they can just go all the way east, and there would be just a sudden shift in the number of species really, really rapidly. So I suppose if that were to happen, then you would see a lot of species richness increase quite drastically. So yeah, but on a very long-term scale, yeah, I'm not quite sure. I would suppose there'd probably be a reduction in quite a long temporal scale. Does that answer your question?
It does. Thank you.
All right, I think people can unmute themselves now if they want to have a question.
Hi, Justin. Congratulations. Hey. So I was wondering, since you had seen these fairly considerable changes in community composition, and you mentioned some implications for food web dynamics in the Arctic, if there is a concerted effort to now collect regular diet data as part of this project. Maybe this is a question for Trent, but are you guys seeking funding to be able to do that because it's potentially so important?
I can't speak for Trent, but it is something that we've talked about. It is not currently part of the project, although the other grad students are doing very similar stuff. I don't think anybody's necessarily doing just or doing specifically diet stuff, but there's a lot of other. So Kyle's looking at otolith stuff, and Duncan's looking at how broad whitefish energetics will affect things. So there's a lot of other things, but part of this is just based on the challenge of the field work. There's just not enough time in the day to collect all these samples with the field crew that we have, and then add on this other major component too. So it's something we would love to do, but we'd have to get another person up there.
The other problem we run into is we try to kind of look at it a couple years ago. In those fike nets, fish sit in there for as long as 24 hours, and so they digest a lot of what they've already eaten. So if you think of some of the fish that are feeding on zooplankton, there's not a whole lot left in their stomachs at that point, or you can't really identify what's there. The other thing that happens is some other fish take advantage of the other fish that are in the net. And so like the sculpins, for example, just gobble up everything they can find. Yeah. So I think it'd be a great idea to do that, but I think the challenge, we'd have to use some other sampling gear like seines or something to get fish in real-time basis so that we could remove contents right there or preserve fish right then. But I think, we were gonna, for example, this summer, if we get to do any field work, we're gonna start collecting zooplankton samples, which no one's ever collected in that area before. So we're just trying to piece together little bits every year. But as Justin mentioned, we're spending all day, or they're spending all day, not me, spending all day up there doing sampling as it is, and to add other pieces in there, you need more time, more people. So we're getting there. It's a work in progress.
Yeah, that's a perfect answer. That's why Trent got to where he is today. He's just good at that stuff. Anyone else have any other questions? Anyone else? Bueller. Well, I suppose if there's no other questions, that's it for today. Thanks everyone for listening in. If you have a question, you can email me later. I'm always open to helping anyone with answering these questions, or where there's just helping them with some of their R code or anything. So thanks again, everyone, for tuning in. And I really appreciate the support from everybody.
Thanks, Justin.